I'm Seth Field, and I'm the Director of Physician Scientist Programs at the Harrington Discovery Institute. It's a real pleasure for me to introduce the next two segments of our program, the Scholar Experience and our keynote address. I'd first like to take a moment to remind you of the depth and breadth of capabilities we've put in place to help investigators advance their drug discoveries into licensable packages that garner commercial attention, which enables entry into clinical practice. Our Therapeutic Development Center, or TDC, includes an advisory team of drug development experts and project managers who work as extended members of each scholar's team to advance their technology toward patient benefit. Our TDC experts have spent their careers in the pharmaceutical industry, and they bring diverse skills in preclinical and clinical planning, pharmacology, biology and biophysics, formulation and chemistry, as well as commercial and business development. Success transpires only with great leadership and skilled project management. In our case, success transpires because of Dr. Diana Wetmore, who is our Vice President of Therapeutics Development and who directs the TDC team and efforts. The combination of advisors and project managers is a winning one that we've developed over the years, and we find it immensely satisfying to hear of scholars who, like our next speaker, have made significant progress in developing new therapeutics. So now, our Scholar Experience presentation. Dr. Ed Grabczyk is a professor of genetics at Louisiana State University Health, New Orleans School of Medicine. He earned his bachelor's degree in psychobiology at UCLA and his PhD in cell and developmental biology at Harvard. He did his postdoctoral work in neuroscience at the Massachusetts General Hospital and in DNA repeat expansion at the NIH. As a 2018 Harrington Rare Disease Scholar, Dr. Grabczyk worked on repeat expansion as it applies to disorders like Huntington's disease and Friedreich's ataxia. Huntington's causes nerve cells in the brain to waste away with symptoms such as uncontrolled movements that typically begin in middle age. Friedreich's ataxia, a priority focus area for the Oxford Harrington Rare Disease Center, typically affects children. These diseases have no cure. Dr. Grabczyk developed a technology designed to slow the progression of diseases characterized by DNA repeat expansion disorders, and in 2023, a Harrington partner, Takeda, licensed the technology. It's exciting work. So Dr. Grabczyk will now tell us more about his scholar experience. Please join me in welcoming him.
So there's a lot of rare diseases, and what people forget when you think of rare diseases is that there's so many rare diseases that it's not rare to find somebody that you know that has one, but it's rare to find a rare disease that has a cure or a treatment. And my primary interest has been Friedreich ataxia, for what Seth was saying, that it affects young people for the most part. And so when you see this type of picture where they have a kid in a wheelchair, if you know the way that this disease runs, it's what's known as a relentlessly progressive disease. So the wheelchair is just a transition. Those children are going to be, they're not going to live into their 30s most likely when they're that young in a wheelchair because of the progressive nature of the disease. And other progressive diseases of this sort caused by repeat expansion are later onset, such as some forms of ALS. Now, at its very most basic level, repeat expansion is that you have a normal gene with a small number of repeats, and then a gene with a larger number. And that's a very simplistic look, because although that explains the difference, what really happens, what really causes the progression, is the constant growth of those repeats over time in the patient. So what it really is, you have this normal range, And then you have a transition range where the repeat becomes unstable and prone to expansion. And then that can go up into the disease range. And it's continuously expanding the entire life of the patient. So it expands so much sometimes that I think of it as like a cancer of the genome. So your DNA is actually expanding and expanding out of control in one location. These are just some of the DNA repeat expansion disorders. Some of the better known ones are, for instance, ALS, and then the one I work on the most is Friedreich ataxia. And so the repeat in Friedreich ataxia is in the intron, and like in junk DNA, but because it expands to such a degree, it suppresses expression of the gene, which then causes the downstream symptoms. Other disorders, such as Huntington's disease, have an expansion in the coding region, so they make a toxic peptide, or toxic part of a protein, that kills the cells that are in which these repeats are expanding. So one of my primary interests is what causes the repeats to expand and what determines the rate. For a typical patient, they get born with a certain size repeat. It expands over their life to cause the symptoms and increase their severity, and then finally, for some patients, end up in toxic levels. Other people are less fortunate, where they get born with a larger repeat. They have a faster disease course, earlier onset, and a quicker death. But there's also people who have the same size repeat as, say, another person, but it expands more slowly. And this is the case for the Acadian Friedreich ataxia population in Louisiana. And so that's the reason I moved to New Orleans to study these Acadians, because they had a slower course of the same size repeat that would predict a much more severe disorder. And one of the things I want to point out to you before, I won't do much data after this one, but this is important for how these diseases run. So you have the normal range, absolutely stable. There's the threshold range, but it's stable in these small numbers, like in the 40s. It takes years to expand, not weeks. But as you get larger, it's now expanding in a week. If you double it again, it expands every day. And so the difference between 44 and 176 is fourfold in terms of length, but it's over 1,000-fold in terms of rate of expansion. And this is one of those conundrums about these disorders. What happens with a lot of them is that the later onset ones, such as Huntington's, you have decades of perfectly normal life. So you might go 30, 40, 50 years with no symptoms. Then the symptoms start, and that's because the repeats reach the point where it's starting to expand faster and entering the toxic area for the particular disease. And as the repeat expands faster and faster, the disease accelerates, reaching an endpoint. So the way I often think of these disorders is that in a given cell, as the repeat reaches its heights, it just explodes the cell. So then you see this kind of thing going on. The repeats are getting to the toxic length and killing those cells. Now those cells are all gone. And you have the downstream pathology. But there's other things that control the repeat expansion besides length, and they're basically DNA mismatch repair proteins. In particular, there's one that's actually the active agent. It's called MLH3, and it cuts DNA in a very specific way that always prefers expansion over contraction. Fortunately, there's a second form called, conveniently, isoform 2 that does not cut DNA. And so I got the idea to simply encourage the formation of isoform 2 to slow down repeat expansion. And it wasn't really moving as fast as I wanted, and that's where Harrington came in. So I applied for this grant in 2017, got it, and then the Therapeutics Development Center has helped me immensely. So we had our first meeting in New Orleans, and they came in and asked me a lot of challenging questions about my aims, about my plans. And it was a little tense for me at first, until at the end of it all, I think it was Diana, said, well, why don't you bring in your team? Let's meet your team. But my team was already in the room because there were only 2 of us. And so this is my team at the time. I had a graduate student myself. And we would joke about our models being very glamorous. And of course, the mouse model is the most glamorous. We gown for doing that. In the summer, my team would expand with a summer student. But for the most part, it was usually just a couple of us. And so accelerating my particular breakthrough first took the form of amplifying my efforts by sort of taking my work and farming it out to other companies that did work for us. And so we got more work done, but more important than getting the larger end is that the pharmaceutical industry likes to see that this type of work can be done by a standard industrial concern, a lab, and it can be scaled. So we scaled it and had other labs doing the experiments that were kind of difficult to do sometimes. And another way that they amplified my, accelerated my breakthrough was by focusing my work. So from the very outset, we refocused my scientific aims. Diana told me one aim just had to go immediately. Another aim died off later from bad results in experiments. And so we kept on refining my approach. And Sue was very instrumental in helping with the lead optimization for these oligonucleotides we were using, testing different chemistries. And finally, one of the very important things was establishing a bridge to industry. Part of this took the form of doing pharma-friendly controls and safety trials, which I would have normally skipped in the lab because I've got a small lab and don't want to do lots of controls. But that helped. And also, I learned to do pharma-friendly presentations predominantly from Steve, who was telling me how to tell the people in a corporation what they need to hear rather than what I want to tell them or what the details of my experiments are. And independent of, or not independent, but on a different angle, on a different side with different people, they also did HC. I did a commercial assessment and development that helped frame this for a pharmaceutical company. And then, as Seth said, Takeda licensed my IP. And so now I finally have the chance to have my dream of actually helping these Friedreich ataxia patients who really don't have much hope. So I'm hoping by using splice switching technology to change the speed of repeat expansion to stop the progression of Friedreich ataxia or stop the actual onset of late onset diseases. Because if we can slow it down even by half, if someone's going to get onset of, say, Huntington's disease between 50 and 60, you can push it out to it's just not going to happen in their lifetime. And for someone like me who's a real procrastinator, putting the disease off indefinitely has a real appeal. Thank you.